Good day, this is Professor Will, CBMD PhD. Today is Monday, June 22nd, 2020. It is 2:58 and 38 seconds a.m. Eastern Standard Time. And today I wrote a little message in addition to my scientific research. So let's get started here. What is a friend? A friend is someone who dares to be there through thick and thin, someone to trust, a shoulder to cry on. A friend is someone who doesn't judge you in 100 different ways that aren't demeaning. A friend who you commend for listening to your dark past. A friend is someone who is compassionate and loves you beyond your defects. A friend spends time with you and makes time with you no matter how busy life gets. One last one, a friend is someone special in your life that makes a difference and wants to see you do good. I have very few friends, but the ones I do have I cherish, and you know who you all are. The second structure of information is used to modify the profile scores to help constrain sequence matching to the structured regions. ProLine is perhaps the most sophisticated and accurate alignment program available. Because of the high complexity of the algorithm, its obvious drawback is the extremely slow computation. Iterative alignment. The iterative approach is based on the idea that an optimal solution can be found by repeatedly modifying existing suboptimal solutions. The procedure starts by producing a low-quality alignment and gradually improves it by iterative realignment through well-defined procedures until no more improvements in the alignment scores can be achieved. Because the order of the sequences used for alignment is different in each iteration, this method may alleviate the great problem of the progressive strategy. However, this method is also heuristic in nature and does not have guarantees for finding optimal alignment. It performs multiple alignment through two sets of iterations, inner iteration and outer iteration. In the outer iteration, an initial random alignment is generated that is used to derive a UPGMA tree. Weights are subsequently applied to optimize the alignment. In the inner iteration, the sequences are randomly divided into two groups. Randomized alignment is used for each group in the initial cycle, after which the alignment positions in each group are fixed. The two groups, each treated as a single sequence, are then aligned to each other using global dynamic programming. The process is repeated through many cycles until the total SP score no longer increases. At this point, the resulting alignment is used to construct a new UPGMA tree. New weights are applied to optimize alignment scores. The newly optimized alignment is subject to further realignment in the inner iteration. This process is repeated over many cycles until there is no further improvement in the overall alignment scores. Block-based alignment. The progressive and iterative alignment strategies are largely global alignment-based and may therefore fail to recognize conserved domains and motifs among highly divergent sequences of varying lengths. For such divergent sequences that share only regional similarities, a local alignment-based approach has to be used. The strategy identifies a block of ungapped alignment shared by all the sequences, hence the block-based local alignment strategy. The method breaks each of the sequences down to smaller segments and performs all possible pairwise alignments between the segments. High-scoring segments called blocks among different sequences are then compiled in the progressive manner to assemble a full multiple alignment. It places emphasis on block-to-block comparison rather than residue-to-residue comparison. The sequence regions between the blocks are left unaligned. The program has been shown to be especially suitable for aligning divergent sequences with only local similarity. The program compares segments of every 9 residues of all possible pairwise alignments. If the similarity of particular segments is above a certain threshold across all sequences, they are used as an anchor to assemble multiple alignments. Residues between blocks are unaligned. The server requires the user to submit a set of sequences in the FASTA format, and the results are returned by email. Alignment at the protein level is more sensitive than at the DNA level. Sequence alignment directly at the DNA level can often result in frameshift errors because the DNA alignment gaps are introduced irrespective of codon boundaries. Therefore, in the process of achieving maximum sequence similarity at the DNA level, mismatches of genetic codons occur that violate the accepted evolutionary scenario, and insertions or deletions occur in units of codons. The resulting alignment can thus be biologically unrealistic. For that reason, sequence alignment at the protein level is much more informative for functional and evolutionary analysis. However, there are occasions when sequence alignment at the DNA level is often necessary. For example, in designing PCR primers and in constructing DNA-based molecular phylogenetic trees. Because conducting alignment directly at the DNA level often leads to errors, DNA can be translated into an amino acid sequence before carrying out alignment to avoid the errors of inserting gaps within codon boundaries. After alignment of the protein sequences, the alignment can be converted back to DNA alignment while ensuring that codons of the DNA sequences line up based on corresponding amino acids. No matter how good an alignment program seems, The automated alignment often contains misaligned regions. It is imperative that the user check the alignment carefully for biological relevance and edit the alignment if necessary. This involves introducing or removing gaps to maximize biologically meaningful matches. Sometimes portions that are ambiguously aligned and deemed to be incorrect have to be deleted. In manual editing, empirical evidence or mere experience is needed to make corrections on an alignment. One can simply use a word processor to edit the text-based alignment. There are also dedicated software programs that assist in the process. In many bioinformatics analysis, in particular phylogenetic analysis, it is often necessary to convert various formats of sequence alignments to the one acceptable by an application program. The task of a format conversion requires a program to be able to read a multiple alignment in one format and rewrite it into another while maintaining the original alignment. This is a different task from simply converting the format of individual unaligned sequences. The bioedit program mentioned is able to save an alignment in a variety of different formats. Multiple sequence alignment is an essential technique in many bioinformatics applications. Many algorithms have been developed to achieve optimal alignment. Some programs are exhaustive in nature, some are heuristic. Because exhaustive programs are not feasible in most cases, heuristic programs are commonly used. These include progressive, iterative, and block-based approaches. The progressive method is a stepwise assembly of multiple alignment according to pairwise similarity. A prominent example is Cluster, which is characterized by adjustable scoring matrices and gap penalties as well as by the application of weighting schemes. The major shortcoming of the program is its greediness, which relates to the error fixation in the early steps of computation. To remedy the problem, T-Coffee and DB Cluster have been developed that combine both global and local alignment to generate more sensitive alignment. Another improvement on the traditional progressive approach is to use graphic profiles as in POA, which eliminate the problem of error fixation. Proline is profile-based and has the capacity to restrict alignment based on protein structure information, and is thus much more accurate than custom. The iterative approach works by repetitive refinement of suboptimal alignments. The block-based method focuses on identifying regional similarity. It is important to keep in mind that no alignment program is absolutely guaranteed to find correct alignment, especially when the number of sequences is large and the divergence level is high. Alignment resulting from automated alignment programs often contains error. The best approach is to perform alignment using a combination of multiple alignment programs. The alignment result can be further refined manually or using RAS. Protein encoding DNA sequences should preferably be aligned at the protein level first, after which the alignment can be converted back to DNA alignment. One of the applications of multiple sequence alignments in identifying related sequences in databases is by construction of position-specific scoring matrices, PSSMs, profiles and hidden Markov models, HMMs. These are statistical models that reflect the frequency information of amino acid or nucleotide residues in a multiple alignment. Thus, they can be treated as a consensus for a given sequence family. However, the consensus is not exactly a single sequence, but rather a model that captures not only the observed frequencies, but also predicted frequencies of unobserved characters. The purpose of establishing the mathematical models is to allow partial matches with a query sequence, so they can be used to detect the more distant members of the same sequence family, resulting in an increased sensitivity of database searching. A PSSM is defined as a table that contains probability information of amino acids or nucleotides at each position of an ungapped multiple sequence alignment. In such a table, the rows represent residue positions of a particular multiple alignment, and the columns represent the names of residues or vice versa. The values in the table represent log odds scores of the residues calculated from the multiple alignment. Construct a matrix raw frequencies of each residue at each column position from a multiple alignment on a first count. Frequencies are normalized by dividing positional frequencies of each residue by overall frequency so that the scores are length and composition independent. The values are converted to the probability values by taking to the logarithm, normally to the base of 2. In this way, the matrix values become log odd scores of residues occurring at each alignment position. In this matrix, a positive score represents identical residue or similar residue match. A negative score represents a non-conserved sequence match. This constructed matrix can be considered a distilled representation for the entire group of related sequences, providing a quantitative description of the degree of sequence conservation at each position of a multiple alignment. The probabilistic model can then be used like a single sequence for database searching and alignment, or can be used to test how well a particular target sequence fits into the sequence group. The probability values in the PSSM depend on the number of sequences used to compile the matrix. Because the matrix is often constructed from the alignment of an insufficient number of closely related sequences to increase the predictive power of the model. a weighting scheme similar to the one used in the cluster algorithm. It uses that downweights overrepresented closely related sequences and outweights underrepresented and divergent ones, so that more divergent sequences can be included. Application of such weighting scheme makes the matrix less biased and able to detect more distantly related sequences. Actual multiple sequence alignments often contain gaps of varying length. When gap penalty information is included in the matrix construction, a profile is created. In other words, a profile is a PSSM with penalty information regarding insertions and deletions for a sequence family. However, in the literature, profile is often used interchangeably with PSSM, even though the two terms in fact have subtle but significant differences. As in sequence alignment, gap penalty scores in the profile matrix are often arbitrarily set. Thus, to achieve an optimal alignment between a query sequence and profile, a series of gap parameters have to be tested. PSI BLAST. Profiles can be used in database searching to find remote sequence homologs. However, to manually construct a profile from a multiple alignment and calculate scores for matching sequences from a large database is tedious and involves significant expertise. Good day, this is Professor Will, CBMD PhD on infectious disease, molecular microbiology, and bioinformatics. I hope you enjoyed listening. Until next time, have a great day.